I welcome you to I'm still your teacher. I'm still your teacher, Charles Batinwarinda. I'm still welcoming you to this next episode, which is where we're going to talk about semi-conservative replication. In the previous video, we were able to look at the different types of replication. Now, let us go straight. How is this semi-conservative replication? Now, we're saying this is a type of replication where a daughter what? Where a daughter DNA molecule, while the DNA molecule formed has one of the strands when it is brand new, and then the other strand when is part or was from the original was from the original DNA molecule when it was from the original DNA molecule. A type of replication where you form two daughter DNA molecules, but each of these two daughter DNA molecules, each one of them has one strand being the original strand, resembling the original DNA molecule, and then another strand being a brand new strand. That's what you call semi-conservative. It means the other one, which was the original, has been conserved, but because it is one out of the two, that's why we call it semi. Yeah. So how does this one take place? We are going to describe this process of DNA replication. We are using, you allow me use this structure of mine. I have this structure of mine, and this structure of mine is our DNA molecule. So I have a DNA molecule which is like that. This is my DNA molecule is like that, and of course this DNA molecule has got these bases, nitrogenous bases, which interact together to what we call hydrogen bonds. So look at this scenario where you have got a DNA molecule which is uncoiled. If I have uncoiled DNA structure, it's like that. Now this structure of DNA structure, I remember we saw when we were looking at the structure of DNA, we saw that the DNA structure is made up of two strands, as you see, but we say these strands were antiparallel. That was in one of those earlier episodes that for those who have watched, who have been following me very closely. Now, it means the antiparallel we saw that it means that one of the strands is running in a 3' to 5' direction. I'm not repeating that because that one I explained it very well in the previous videos. And then another one is having 5', 3', is running in a 5', 3' direction. Now, these are the two strands that we are having. Now, when we are looking at in the recent video, we saw that we have got enzymes which we need for us to be able to carry out replication. One of the enzymes was an enzyme which was called DNA helicase. DNA helicase. Now, here it is at this point where DNA helicase will come, help us do the work. The DNA helicase does which work? The DNA helicase will come and help us unwind this structure, this DNA molecule, unzip this DNA structure. How? It will unzip. I told you that these hydrogen bonds you see here, it's as if they are like, take them as zips of somebody's trouser. So when you say unzipping, it means opening the zip. So it means these bonds must be broken down. They must be broken down, and then when they are open, it means you would have unzipped, you would have unopened, you would have these ones are together, you would have opened them up like this. Yeah, so we have got our DNA structure, we have got our DNA helicase which will come. Now take this structure of mine, take this enzyme of mine here as my DNA helicase. Take this one as my DNA helicase enzyme. This DNA helicase enzyme is the one which is going to help us to, and the work of this DNA is to unzip. unzip the DNA molecule, How? By breaking the hydrogen what? By breaking the hydrogen bonds. These hydrogen bonds are the bonds which are holding the two bases, the bases of the two strands, of the two adjacent strands together. The hydrogen bonds are these ones here. These are the hydrogen bonds we're talking about. So the DNA helicase will catalyze a reaction which will help us to break those bonds apart, so that the DNA strand which was connected together so that the DNA molecule which was joined together, but where the two strands were joined together, can separate. So the DNA helicase, so this process of the process of DNA replication, so the process, if I can write here, we are saying the process begins with what? With the DNA helicase enzyme attaching itself at what? At one end of the DNA what? Of the DNA molecule. And when it attaches itself at one end of the DNA molecule, what does it do? This DNA helicase, the DNA helicase enzyme will speed up the what? The breakdown, the reaction which involves the breakdown of what? Of hydrogen bonds. And when these hydrogen bonds are broken down, then this will cause the unzipping. This will cause the unzipping of the what? This will cause the unzipping of the DNA molecule. And when the DNA molecule unzips, it unzips forming a Y shape. Also called replication what? Which also called replication fork. Yeah, a fork, a fork has got some, a fork is something like this with some, some finger-like structures here. Yeah, so we are saying once these bonds are broken down by this helicase, once these bonds are broken down by this DNA helicase, what happens is you form the DNA opens up and it forms something which is like a Y, shape. What do we mean? We are saying once this DNA helicase has moved around and has kept on opening up this DNA strand, you will end up forming a structure. Which is like this. So this bond has been broken. So you end up having something which is like this, like that. So what do we mean? So it means this one has been broken, and we are still breaking more. So it means here we still have other bases which have not been, where the bonds between them has not been broken down. Here, like that. The bonds there have not been broken down yet. There the bonds have not been broken down yet. Then here, these bonds are still not yet broken down. So it means the DNA helicase, for example, most likely DNA helicase has just reached here. It has finished breaking the bonds which were holding all these bases which are this side. So it means here, where we have broken, we have got now bases which are just exposed. We have bases which have been exposed, which have been separated like that. We have bases which have been separated like that. Likewise, also down here, you also have bases which have been separated. Remember, each of this one has where it was attached down here, like that. These are all the bases which have whose hydrogen bonds have been broken down by this DNA what? By this DNA helicase. So this is our DNA helicase, which has broken hydrogen bonds which were holding all these bases together for the two strands. Mind you, we have said that one of these structures up here is this strand here, which is on our part. We have assumed it is the one which was where the carbon atom number 3, the carbon atom which at the extreme end is the carbon atom number 3. We're calling it 3', and then this one is 5 prime. The carbon atom which is at this extreme end is a carbon atom number 5 on the pentose sugar. Down here, and we have said because the two strands are antipyrable, it means this one here. If this is 3 prime, 5 prime, it means down here, this will be 5 prime, then 3 prime, like that. Yeah, so we are saying, for during this hypothesis, we have begun by describing what is DNA, what is semiconservative replication, where one of the DNA molecule is broken down to form two DNA molecules, but each of these two DNA molecules, one of the strands is the original, and another strand is brand new. We have seen that for DNA replication to take place, we begin with the DNA enzyme, which is called DNA helicase. This DNA helicase moves along the DNA molecule, and as it moves along the DNA molecule, it moves while unzipping the DNA molecule. How does it unzip? It unzips the DNA molecule by breaking the hydrogen bonds which were holding the DNA molecules together. Now, after that has happened, we have seen that you form a structure like a Y. I hope you are seeing, actually, this one is really looking like a Y. This is why the Y we are talking about. See, this is something which looks like a Y. And we are saying that Y structure, we are referring to it as a replication fork. Now, after we have understood that, we are saying that the two strands of the DNA which is undergoing replication are oriented differently. How? Because they are antiparallel, one of the strands runs in a 3 prime 5 prime direction, while the other one runs in a 5 prime to 3 prime direction. Now, what are these strands here? This strand, the strand which is okay, so for those ones who are noting, for some of my friends there who told me that when you are watching, you watch as you are noting down, let me give you something to note down. For those friends of mine who say that when you are writing down, when you are seeing, you keep noting down, is after that one we have said that the strand, the DNA molecule, which has unzipped is antiparallel. How? That is to say, one of the strands runs in a 3 prime to 5 prime direction, while the other runs in a 5 prime to 3 prime direction. Now, we are going to say that the DNA structure, the DNA strand, which is running in a 3 prime to 5 prime, like for example, in this case, this strand, which is on the upper part, that is running in a 3 prime, 5 prime, remember, this DNA helicase is moving in this direction, is moving the other side. So this structure, which is moving in a 3 prime, 5 prime, with respect to DNA helicase, how it is with respect to how DNA helicase is moving, that strand, we are referring to it, it is given a name, and that strand is called the leading strand. The leading strand. We call it the leading strand. Hey, please take note of that seriously. That structure, that strand, which runs with respect, of course, we are naming these strands with respect to how this DNA is opening. Now, if the DNA is opening like that, we are saying the strand, if the DNA is opening, moving in this direction, this strand, which is with respect to the movement of the DNA helicase, take note, I'm saying with respect to how DNA helicase is moving. The strand, which is in a 3 prime to 5 prime direction, that is our therefore means this is our leading what? This will be our leading strand. That's our leading strand. Then the other strand, which is moving in 5 prime, I am naming starting from the direction where the helicase is coming from. Please take note, I am naming starting or basing on where the DNA helicase is coming from. That's why I'm saying with respect to DNA helicase, how it is moving. So with respect to the movement of DNA helicase, the strand which is moving in a 5' to 3' direction, that one is called the lagging strand. The lagging strand. You know the meaning of the word lagging? When something is lagging, it means it is coming after the other. When something is leading, it means it is going ahead of the other. So we are saying, and there's also a story we are calling them like that. It means one of the strands is doing first, another one is coming later. Now, somebody might be confused by this statement of saying 3' to 5' direction and so on. It means now, let me first bring you here. It means if I have my DNA, now here I've drawn it like this, but assume I had drawn my DNA structure without rubbing this. Assume I had drawn, let me first use something small here. Assume I had drawn my DNA structure like this, and my DNA helicase was moving from here. Okay, this was 3', 5', then this is 5', 3'. But now the helicase was moving from this side, moving this side. If I may ask you, this is the DNA structure, but this is the strand A and strand B. Strand B and strand A. Now, if I may ask you, because whenever I'm teaching this in class, I usually find some students getting lost, especially when you talk about 3' and 5', and then leading and lagging. Now, I have said, with respect to where the DNA helicase is moving, if it's moving from this side to this side, now look at the strands. The one which is in a 3' to 5' is the leading strand. Then the one which is 5', 3' is the lagging strand. So that's why I've called this one the leading, and then you are calling this one the what? I'm calling this one which is down here the what? The lagging strand. It means the one which is going to be stranded the last. It is where they to be stranded the last, to come last in the strain. Now, if we have understood this one, let me check you on this. If I have this DNA as my DNA molecule, and I am saying this is the DNA molecule, and now helicase is moving in this direction, is moving from this side to this side. I've maintained it is just like though you see this one, the way I had this one, I just didn't want to rub this. I wanted to just first leave this one here. Now, I'll remain with this same molecule, but now the difference is that the DNA helicase here was moving like this. But in this case, DNA helicase is moving in this direction. If it is moving in this direction, I have strand A and strand B. According to you, which one do you think is now the one which is supposed to be the leading strand and lagging strand? I'm asking you. Which one do you think is supposed to be a leading strand? Is it strand A or strand B? I don't know what is moving in your mind. But if you have said it is strand A, it is strand B. If you have said that if in your mind you are saying it is strand B, is it the case? Is it the case? If you have said it is strand B, then you are correct. You have understood the concept here. So it means if the helicase is moving like this, opening like this, so it means I will end up having a structure after the helicase has finished opening. I will end up having a structure which is the reproductive fork is going to be like this. So I have a 5' here and a 3' here. The helicase has so far reached here. This is 5', 3'. Now I've said with respect, the helicase is moving this side. Yes, I'm saying with respect how helicase is moving, with respect how the helicase is moving, I am saying that the DNA strand which is in a direction of 3', 5' is what we are referring to as the leading strand. So it means in this case my leading strand will be this one. This will be my leading strand. Then this will be my lagging strand. The upper one will be the lagging. The one which will be down will be the leading. Why are we calling it leading? We are going to see why you are calling it the leading strand. But we are the concept as for now. We are saying the one which is moving with respect to how the DNA helicase is moving. With respect to the direction here, with respect to the direction of the movement of the DNA helicase, the strand which is moving in a 3' to 5' direction is called a leading strand, while that one which is moving in a 5' to 3' direction is what we are referring to as the lagging strand. I am saying a keynote here that with respect to how DNA helicase is moving. Okay, now, because of this, when you check, we are saying the leading strand and lagging strand. Because of this, we are saying these strands are going to be oriented differently. Why? Because these strands are oriented differently. Because we have got different orientation of these strands, whereby one is moving in 3' to 5', another one is moving from 5' to 3'. I've explained 3' and 5' in the previous videos. For those ones who have been following, thank you very much. Those ones who have not been following, don't get lost. Just first go to those previous videos. Now, because these strands of the DNA molecules undergoing replication are oriented differently, therefore, their replication, the way they are going to replicate, the way they are going to form new copies, is also going to be different. Why? And we are saying that each of these strands, the strand which has been formed and this strand which has been formed, are the ones which are going to act as templates. They are going to act as templates. Now this one is going to be a template. This one is going to act as a what? As a template. A template is something you use to make another thing. A template means something you are using to make something else. So these ones are going to be acting as templates, which are going to be used to form another new molecule of DNA. So we are saying from here that because we are saying after that statement, for those friends of mine who told me that you like writing. Now after that statement, we are saying because the strands are oriented differently. It means they are oriented differently. It means even their replication is going to be what? Is going to be different. It's going to be different. So because we are saying, because someone might say, I'm not seeing what you're writing here. Those are not writing, who are not seeing what I'm writing here very well. What I'm writing is what I'm speaking. Because these strands are oriented in different ways. They are not the way they are orienting is not the same. The way they are organized, the way they are flowing is not the same. One is facing one direction, one is facing in the other direction. Because they are oriented in a different direction, even their replication will be different. Now, I want to know, how are they replicated? Now, this leading strand is going to be replicated in a format that we call continuous replication. Why don't you join me in the next episode? First, we can expound on that. Thank you very much.